Microbial interactions with root systems and soil animals, resultant effects on soil respiration by James Gower. Soil respiration is a major contributor to global CO2 production. In fact, soil respiration is estimated to be 10 times that of man-made CO2 production. Small disturbances in soil respiration can have a powerful impact on atmospheric CO2 levels. Despite extensive study, the exact nature of the interactions that dictate soil respiration are still not fully understood. Interactions between soil CO2 sources have been shown to be key to understanding soil respiration. Mycorrhizal interactions between respiring plant roots and soil-bound fungi have been shown to augment soil respiration, as have interactions between soil animals and their symbiotic microbial neighbours. In light of the research carried out in this area, an experiment was designed to explore the significance of interactions in determining soil CO2 efflux. The experiment sought to examine the effect of mycorrhizal activity on soil CO2 efflux, as well as the significance of earthworm microbe interactions occurring in the drillosphere. The hypothesis prepared for the experiment was that CO2 efflux generated by soil CO2 sources, such as plant roots and soil fauna, is significantly augmented by the presence of soil-bound microbes. Ultimately, the experiment hoped to determine the importance of intersoil interactions, as well as assessing the relative value of further research in this area. The soil used in the experiment consisted of a mix of mineral-rich clay soil and Verve high-quality commercial organic compost, mixed at an even 1-to-1 ratio. The mineral-rich clay soil was collected from a local community garden. After thorough mixing, the soils were autoclaved to achieve sterilization. The soil was then divided into 30 pots. Half the pots were then inoculated with microbes. The remaining 15 were left sterilized. The pots then received either fescue seeds, earthworms, or no treatment. This ultimately divided the pots into 6 treatment groups. While in the lab, the soil was exposed to 24-hour lighting and regularly watered with distilled water. After 2 weeks, measurements were taken over 12 days at 3-day intervals. This involved placing a closed chamber over the soil pot and using an infrared gas analyzer to determine the change in CO2 concentration inside the chamber headspace. CO2 efflux could then be calculated using the formula F equals SV over A, where F is CO2 efflux, S is slope of CO2 concentration versus time, V is chamber headspace volume, and A is the area of the soil. Statistical analysis of the CO2 efflux readings was carried out using 2 statistical test types. 2-sample t-testing was used to compare sterilized soil pots exposed to secondary treatments with their various analogous inoculated equivalent, as well as to compare sterilized inoculated pots with other sterilized and inoculated pots that differed only in secondary treatment types. A 2-way ANOVA was used to determine if interaction occurred between primary and secondary treatments. Here we can see a graphical representation of the CO2 efflux readings for inoculated and sterilized soil treatments. The graph depicts an increased level of CO2 efflux in inoculated soil over sterilized soil across all treatment types. These graphs show fescue and earthworm treatments compared against their sterilized and inoculated counterparts that received no treatment. Finally, this graph depicts the CO2 efflux increase that occurred in all 6 treatment types as the 12-day experimental period went on. The results indicated that considerable interaction was occurring between fescue and microbial components in the soil, the result of which was considerable augmentation of CO2 efflux in pots in which fescue and microbes coexisted. However, by contrast, the results did not indicate that the co-occurrence of earthworms and soil microbes caused augmentation of soil CO2 efflux. There were limitations on the experimental design. In many ways, the soil conditions impeded earthworm proliferation. The absence of organic leaf litter on the surface of the soil impeded earthworm feeding activity and may have stunted their impact. Continuous lighting was employed to suppress earthworm migration and supply fescue with a steady supply of light for photosynthesis. Continuous lighting, however, can be damaging to plants and trigger oxidative stress. Similarly, the loss of the day-night cycles is likely to have impacted earthworm feeding behavior and disrupted their respiratory contribution. In conclusion, the study did confirm the importance of mycorrhizal interactions on soil respiration. The experiment did not confirm the impact of fauna-microbe interactions on soil CO2 efflux. This was surprising, as the positive effect of fauna on soil respiration is well documented in ecological literature. In nature, fauna, root systems and microbes of many species interact in a complex network to determine soil CO2 efflux, along with abiotic factors. An all-encompassing model is badly needed, and as a result, further research to garner a more sophisticated understanding of soil respiration is advised. Thank you for listening.